So as you can see, it's a very bold title. I think 125 experimental heterogeneity. Well, I did my PhD at Université Côte, and today I'll be talking about synaptic plasticity. So to motivate us, let's see if it's changed. Okay. So neuroplasticity, it's what allows this boy under a very specific stimuli to become this man. So that give names to this institute. So if you provide for the brain of this young boy that's not so different from ours, we likely will have an outcome of this guy. But we agree that it's a very hard experiment to control because we cannot make sure that we're giving to each pair of neurons the same stimuli. So if you want to reproduce this, that would be very hard. Let's try to take a more sample example and just look for a pair of neurons. And you still can see that's quite complicated yet. So this is an experiment done in 78. It was one of the first experiments to show frequency-dependent plasticity. So if you give for a pair of neurons a very fast stimulation, like 100 Hertz, you have a positive change in the EPSP amplitude. So if you give a very low frequency, like 1 Hertz, you make the neuron to decrease this EPSP amplitude. We call a positive change LTP, long-term potentiation, and LTD, long-term depression. So what happens, what is heterogeneous, if you take this example of 1 Hertz and change the age of the animal, there's a screen there, okay. You notice that the LTD is not the same given the age. So that's why we call it heterogeneous. It happens that different laboratories, they have different methods of making these experiments. They change the temperature, the age of the animal. So even if we consider just a pair of neurons, this experiment reproducing the same changes in the connectivity of neurons, this string of neurons is really complicated. Just check the time here. Okay. So because of that, I tried to model with a high precision the largest source of calcium that causes the changes in synaptic plasticity. So I tried to incorporate these heterogeneities between laboratories that consist in age differences, temperature differences, and ACSF differences. ACSF is the liquid they put the slices of brain, so they have different recipes. So it's a very complicated model. So for instance, in the NMDA, you have different subtypes towards the age of the animal. It will change. One NMDA is fast, another one is slow. Sometimes experimentalists, they do experiments in different temperatures. They like to use very cold experiments because the slices, they will not get rot so fast, so they can work for longer. But it's very far from the dynamics that we expect to see in physiological conditions. So also incorporate temperature sensitivity in the NMDA receptor. And also I incorporate the effect caused by different concentrations of ions outside the cell. So if you have a high concentration of calcium outside the cell, the NMDA will restrict the entry of it. So if you have a little bit, the NMDA will allow more calcium to enter. So given that we have a quite detailed model of calcium that's likely to predict plasticity, we went to see how can we use calcium signals to predict the outcomes, long-term potentiation or depression. The textbook says that, the classical approach says that if you have a high amount of calcium, you generate LTP. Let's see where's my mouse, okay. If you have an average amount, you can have long-term depression. And if you have this under this threshold, no change. And another hypothesis to how calcium changes plasticity is called the duration hypothesis. That says that a very long and sustained amount of calcium generates LTD, and very brief and high amount of calcium generates LTP. So because this strategy did not achieve a great coverage of the literature, we went to a little bit deeper the calcium cascade. hate, right? Because when calcium enters in the cell, it binds to other molecules. And we went to look for these molecules, which one controls long-term potentiation and long-term depression. We found out 2 key molecules. One is called KMK2. When you block it, you don't let LTP occur. And another one is called calcineurin. When you block it also, when you inhibit, you prevent LTD to occur. So there's an experiment showing that when you inhibit calcineurin, you don't have LTP. When you inhibit KMK2, you don't have LTD. Okay. So in the end, we came out with this very complicated model. It has it's a stochastic model of a dendritic spine. It has 100 equations and 100 stochastic transitions. I know that mathematicians, they hate this kind of thing, right? They want something more amenable to their methods that have 2 or 3 variables to apply their nice methods. So we went to this complicated model to a reduced form of it. What I mean is we just took 2 variables here and tried to say everything that we can of how the dendrite will change the plasticity, just looking for these 2 variables. So it's kind of a readout, because when you have a complicated system, you want to interpret it. So a readout helps do that. The classical readout says that you can take a threshold and apply to your dynamics, and everything that's above that will give your answer. Okay? The problem of that is sometimes what is important for you is taking consideration with also what is not important. So if you threshold your dynamics, sometimes you take everything in consideration, relevant dynamics and irrelevant dynamics. So what I want to try is to just the dynamics that matters for me, to classify to become LTP or LTD. So for instance here, this threshold cannot make this separation, cannot take this segment of the dynamics that I want. What can do that, it's a more flexible strategy, is this readout. Putting a square here implements exactly this selection that I'm showing this part. So using this strategy that I call geometrical readout, we took 3 experiments and we find out that if I put the combined dynamics of this calcineurin and KMK2, these 2 molecules coding for LTP and LTD, I managed to tell which dynamics will give me LTP or LTD. How I can do that? If I stimulate and this orbit crosses the LTD region, I have a region indicator that will change according to how this orbit will travel across the space. And in the end, to tell me the precise amount of synaptic change that I have this plasticity Markov chain that starts with 100 states here, and will jump for LTP or LTD given the amount of time it spends in each region. So just to show how one example of LTP, one example of LTD. This is an experiment that no other model can reproduce. We test previous models from like Nicola Brunel model, Claudia Clopin model, and this is the only model that can reproduce this kind of experiment. So this is an LTP example. You see the orbit spending some time there, and here you have the threshold activating, and then you have the LTP outcome. But for LTD, you have a different temporal requirement. You need to spend a longer time inside the LTD region. So it's a little bit longer. That's why when you cross this region, when you're coming back, you don't have LTD because you need to stay a long time inside this place. You see, even if you have LTD here, it does not budge the weight change. All right, so this is an example of how this model works. So first we had a very complicated model, and then we find a readout to interpret this complicated model. What is good in that is that we can reproduce the variability of this experiment that is an FDP experiment. So here we have a causal and anti-causal, and what you expect is when you change the causality, instead of having LTP, you have LTD. But you see that's not the case, and this is one of the reasons or the models cannot reproduce this experiment. And if you remember the first motivation that I was showing age-dependent plasticity, that in adults, the LTD is quite weak, we also can reproduce that. The model can capture this aspect. And that's the first model that can capture age-dependent plasticity. Really nice. I like it, at least. So with this model, also, we can try to tell how in vivo-like plasticity rules are. For instance, we know that very young animals, here's 5 days old, they're a little bit colder, so we can set the temperature and see how, if you set a couple two neurons together, one with a postsynaptic rate, another one with presynaptic rate, we can see how the plasticity will be. So here we see that there's no plasticity almost. And once you start to get older, like 15 days and 35 degrees, that's the temperature for this animal at this age, we see that a window starts to form. So we can try to estimate how in vivo-like rules will be. Because they already show that this classical STDP rule, spike time dependent plasticity, is not observable in vivo. So if you try to take data and fit to this rule, we will not see this relationship. What is a problem? Because a lot of models use STDP. In STDP, it's mostly used to justify that your model is biologically plausible. So that's a problem, because when it was done, the STDP rule, it was done in a very cold environment for embryonic cells, embryonic neurons. So it's quite not realistic, and everyone uses it as realistic. So take care. All right. So just in a summary, I think I have a little bit of time. Yes. Oh, for 2 minutes. This model can give us insights about just changing some parameters of the experimental conditions. You can have an idea of how the orbits will be. So here I have 35 degrees. And if I decrease a little bit the temperature, because the dynamics will be a little bit slower, the orbits will be reduced because you have a little bit more of calcium entering. So if I increase the temperature, these orbits will be like that. And also this model is a stochastic model. And if you remove the stochasticity, we made a version of it, it will be not the same. So we need stochasticity. And also, it's really nice that this model is, I'll say, break the record of experiments reproduced. I don't know if this is important or not. But it uses only one readout. You don't need to change the readout to reproduce 10 experiments at the same time. And big models like the ones used by the Blueprint project, they have this version of a model that needs to change the readout all the time to reproduce different experiments. So with this model, we can reproduce without changing the rule. Something that's also good that you can provide for experimentalists, also something that's quite important as testable predictions. Like they need, for instance, a fast way to see if their protocols is doing well, because otherwise they have to spend many animals trying to get the right amount of plasticity change. And this is a way to give them a smart start. You can say, try this protocol with this temperature and stuff like that, and they can do it faster. So it saves their time, and it's really good for them. And so you can improve the experimental design. You can recover the LT that I showed before that's lost for adult animals. And it's the first plasticity model that can capture age, temperature, and extracellular medium at the same time. And that's it. I think finishing 15 minutes. That's great. And those are my collaborators. And right now, I start to do experimental neuroscience in the Marvel Lab, University of Sussex. So I'm kind of getting out of mathematics for a little bit. And that's it. Thank you.
Thank you, Yuri, for a perfectly timed talk.
Yeah, it was quite a...
Any questions from the audience?
Yes. He was my teacher in Lascaux, a long time ago. Thank you.
Thanks for this interesting talk. Just a clarification, when you showed the changes in concentration of campeinase 2 and calcineurin, you're referring to the bound form, the calcium bound form of two molecules.
Okay, you want to know if...
On slide 7, when the first time you're showing us the map.
You're saying this place? This?
No, the one before.
Oh yes, yeah.
So when you write the concentrations, that's the bound form of these molecules. So that's the calcium bound form.
If this is the calcium boundary?
No, that's the calcium bound form of the molecules.
If the calcium bounds for the molecule, yes, it does. Actually, KMK2 captures calcium and calmodulin, but the calcium binds to everything. Is one of the main messengers in the cell, so it binds to everything. So the thing is, if you block one of these molecules, you cannot see the compensation that the other molecules will be having, because they're more calcium to bind if you block one. So this model cannot reproduce inhibition of these molecules, because you have many others.
Yeah, thanks. And another question: when you're saying that model is stochastic, you're working with concentrations for these molecules, or you're working with?
Is the concentration is we have data that matches perfectly, or data change the temperature, looking exactly to the dendritic spines and accessing the changing bright of these molecules. So it's not concentration, but it's the changing bright.
Okay, but my point is you're not modeling the individual molecules.
No,
your work. Okay,
it is not just the concentration. That's correct. Yeah.
More questions? Go and collect some online, but I have one question for you, Yuri, in the meantime.
Yeah.
So I know you mentioned that in some sense this is a hugely complicated model that you're looking at, and you have a simple readout.
Yes.
So from a viewpoint of, so you will have operated obviously this model many times, and I wonder if you can single out some constitutive components that help you at least stripping your 100 equations into something more manageable. I'm not trying to say, oh, you need a simpler model, but I would like to know from your experience, what are really the fundamental things to obtain that readout?
What are the fundamental things?
You really need the entire 100 models and 1,000 parameters.
Ask me to pinpoint one variable. That's the one that
some,
change.
yeah, or some mechanisms that are more important to get that readout.
Yes, I can say one that's really, really important, because when you're making a deterministic version of this model, we change from stochastic to deterministic to see what matters, right? Mm-hmm. And we noticed that the most important part of stochasticity, that ones that will quite modify the shapes, is the baseline activity of calcium. That's given by the flickering of calcium channels that stay always open and closing, even if they don't have any stimulation. So one very important parameter to have this done right is the calcium channels that will open even if you don't stimulate, because they'll give the static behavior of your model. Once you have this right, then everything else is not so hard.
Thank you. One question from Emre on YouTube. Did you ever consider also orbits taking into account the temperature change dynamically in terms of temperature during the calcium intake and or with increasing and decreasing activity?
I didn't, but that's an interesting question, because if you stimulate with the 100 hertz, you generate some heat. And certainly the temperature will be not the same. But also we did something very cool, that is trying to inverse the problem of calcium dye, because when you are measuring the calcium, one of your possibilities is using the calcium dye, but for you to see the calcium dye in the slices, you need to shoot some laser, and the laser will increase the temperature. So you have to take this into account to have the real dynamics of calcium. Yeah.
Any more questions before we move on? Let's thank Yuri again for a very interesting talk.